Hi there, welcome to lesson 7.6. This lesson covers the ecology of fungivorous or fungal-feeding nematodes. We'll look at some more examples of fungal-feeding nematodes and fungi interactions, examining various ecosystems and how fungal-feeding nematodes behave in these ecosystems and compare some of the fungal-feeding nematodes, their response to earthworms, as well as their response to cover crops, manure additions, and reduced tillage. The image on the left is a scanning electron micrograph of Aphelinchus aveni, demonstrating its unique anatomy. Okay, so let's jump into lesson 7.6. So let's look at the ecology of some of the fungal-feeding nematodes. There was an honours project conducted at the University of Adelaide in 1984, and two different genera of nematodes and various fungi were isolated from a wheat field and a pine forest soil. Aphelinchus aveni was predominantly found in the wheat field, and Aphelinchoides species was abundant in the pine forest soil. Both genera of nematodes are generalists, but their presence depends on the plant community and the fungal community that they feed on. There was a great variation in the genera of fungi as their food choices, as well as the different fungal species groups, and also the variation was based on individual fungal isolates as well. So the nematodes either preferred certain genera of fungi, certain groups of species, or actual individual isolates of fungi. Both nematodes were supported by the same fungal isolates, so that means that both of the nematode genera preferred the same fungi as a food source. There was more of Aphelinchus aveni in terms of nematode biomass as opposed to Aphelinchoides species. So basically there were more Aphelinchus aveni as opposed to Aphelinchoides species. The greater fungal diversity in the wheat field soil supported Aphelinchus aveni presence in that soil, but these nematodes were actually rare in the pine forest soil, and it was proposed that perhaps it was the competition in the pine forest soil that would have not allowed Aphelinchus aveni to proliferate, or maybe it was predated upon by other nematodes or other organisms more frequently, as well as the fungal foods that it prefers to consume might not have been present in that soil. So this just demonstrates the variability between the different fungal feeders, the different genera, and whether they persist in particular soil types based on the food sources that are available, as well as on their speed of growth or their own life cycle length, as to whether they can be supported in particular soils, and whether there are predators present in the soil that prefer that specific species. So it really just demonstrates the variability and specificity of fungal feeders' prevalence in particular soils. Here we have an image of the genus Aphelinchoides. Another interesting study investigated fungal-feeding nematode and fungi interactions on nitrogen mineralisation in subtropical rice paddy and wheat soils. So the researchers isolated Aphelinchus aveni from that soil and two different species of fungi. They named the fungal isolates F1 and F2, and then conducted these different treatments in microcosm experiments to see how the interactions between the nematodes and the fungi affect nitrogen mineralization. The treatments included soil plus fungal isolate number 1, soil plus fungal isolate and the fungal feeding nematode, soil and fungal isolate number 2, and then soil and fungal isolate number 2 plus Aphelinchus aveni nematode. Both Aphelinchus aveni numbers and fungal growth were increased when both organisms were present, and that's the case for both the fungal isolates. Over the first 12 days of incubation, there was an increase in the density of fungal isolate number 2 by 2.5 to 3.5 times, while isolate number 1 increased in density after 12 days, and that density was increased by 5 to 5.7 times. The nematode numbers increased over the initial 20 days and then slowly declined. Aphelinchus aveni numbers were greater when feeding on fungal isolate number 2 versus fungal isolate number 1, and the presence of nematodes increased soil ammonium compared with treatments where only fungi were present. Fungal isolate number 1 had a greater effect on nitrogen mineralisation in comparison with fungal isolate 2. So this study clearly shows that Aphelinchus aveni prefers different species of fungi once again. Different fungal isolates, different fungal species have different growth response to the soil they grow in, as well as to their interaction with the nematodes. And this translates into how these different isolates, as well as their interaction with the fungal feeding nematode, affect the nitrogen cycle in the soil, nitrogen mineralization, and then therefore the availability of that nitrogen to the plants and to the soil organisms. So the bottom line really is that once again we are going back to this absolute necessity for having biodiversity in the soil. And this is obviously a very simple system where only 2 fungi were isolated and tested with Aphelinchus aveni, so 1 nematode. Obviously, in the soil ecosystem, there are many more different nematodes, many more different fungi, and many other variables, such as soil moisture, soil organic matter presence, and other conditions that will influence these interactions. So it is always important to remember that biodiversity is key, and of course, the fact that there were only 2 species of plants present in the soil will also affect the ecosystem. So the biodiversity of all components of the ecosystem is important, and this will create the greatest potential for plant growth when that biodiversity is allowed to flourish. I must mention that I couldn't find any information on these 2 fungal isolates, and Moniliaceae seems to be a family rather than a species, so maybe it was an isolate from that family. And I've only summarized the abstract from this study because the rest of the article was actually in Chinese, which I couldn't read. So there are no images of these 2 fungi that were named in this study, and I've exhausted all the Aphelinchus aveni images from the web. So due to lack of images to be used for this slide, I've looked up rice paddy and discovered that Chinese farmers are actually producing big images in the rice paddies in their rice fields using various varieties of rice that produces different colors and almost a 3D sense to their images. It's quite an incredible effort on their part. And in this particular case, this artwork is related to a TV series called The Legend of Mi Yue. And there's various different types of artwork, so you can have a look at the beautiful creations of all different themes that these farmers present using different varieties of rice. So this is the sort of stuff that happens when you're creating these sort of lessons, and sometimes there isn't the appropriate image, but there is always something of interest that can be used, and there is always something new to learn. Another interesting study showed the effects of fungal-feeding nematodes, arbuscular mycorrhizal fungi, and organic pig manure on uptake of phosphate and plant growth. It was found that predation increased fungal spore germination and radial avascular mycorrhizal fungal mycelium growth. So basically, the fungi proliferated better around the roots when fungal-feeding nematodes were present. And it was also found that there was an increase in digested avascular mycorrhizal fungal fragments in Aphelencoides and Aphelincus, with an increase in manure amount added to the soil. So the manure addition actually promoted the feeding of the fungal-feeding nematodes upon avascular mycorrhizal fungi. Aphelencoides and Aphelincus also increased glomus external hyphae. So that's sort of a similar statement to that first one, where the avascular mycorrhizal species showed a greater abundance of hyphal growth around the root zone, around the rhizosphere, when the nematodes were present. Aphelincus species showed a greater rate of feeding and therefore more impact on the abundance of the avascular mycorrhizal fungi as opposed to Aphelencoides. So it means that Aphelincus actually stimulated this positive effect of its grazing on the avascular mycorrhizal fungal proliferation. The question the researchers asked was whether the avascular mycorrhizal fungal development was increased due to the recycling of minerals from the senescent fungal tissue. Another option they proposed was that the nematodes preferentially fed on non-active avascular mycorrhizal fungi and therefore releasing those minerals from the fungal tissue, and or consuming other fungi that would compete with the avascular mycorrhizal species. So therefore this means that the avascular mycorrhizal fungal proliferation or increase in the presence of nematodes would be due to the fact that the nematodes were consuming fungi that would actually compete with the avascular mycorrhizal fungi, as well as consuming the non-active senescent avascular mycorrhizal fungi, so therefore not really consuming the new growth or the fresh active avascular mycorrhizal fungi that were growing. They found that fungal-feeding nematodes therefore modulate avascular mycorrhizal fungal community functionality, and this caused a positive plant avascular mycorrhizal fungal interactions. They also found that when nematodes grazed in the rhizosphere, this increased soil alkaline phosphatase activity, so it's an enzyme that allowed for the mineralisation of the fresh organic pig manure that was added to the soil, and this increased phosphate cycling, solubilisation, uptake by the plant, and therefore increased maize productivity. The images on the right are taken directly from the study, and they show the avascular mycorrhizal fungal vesicles as well as avascules in the plant cells. Here is an avascule and the fungal hyphae growing throughout the root cells. This was one of the mechanisms they used to measure avascular mycorrhizal fungal abundance by counting these avascular mycorrhizal fungal structures within the root after staining these structures. So this is just another example of an interaction between the inputs that can be made to increase the nutritional content in the soil and the organic matter in the soil, as well as the symbiotic fungi that are essential for plant growth, and how fungal-feeding nematodes interact with these two components and have an effect particularly on phosphate uptake and therefore in stimulating and supporting plant growth. This study examined the effects of cover crops on nematode populations, and this research was done once again in China in tea gardens. They compared various treatments in the tea gardens where they examined the natural cover that was on the ground and then added 2, 4, or 8 plant species as the various treatments. And these were the cover crop treatments. And they found that cover crops led to changes in soil organic matter, pH, and phosphorus content. Increase in cover crop diversity increased nematode abundance, particularly bacterivores and fungivores, so fungal feeding nematodes, and decreased plant parasite numbers, so nematodes that feed on plant roots. With humic acid accumulation, there was an increase in bacterivore and fungivore populations. Cover crops also were seen to increase organic matter decomposition, and that's because lots of the plants would have actively dropped plant material, and that would have increased populations of fungi and bacteria in the soil, depending on the type of cover crop that was grown. So it was found that plant functional groups, so for example grasses versus forbs versus legumes, have an effect on the composition of soil nematode communities. And the nematode communities are dependent on the microbial populations. So abundant, easy to decompose organic matter is more likely to be available to bacteria. So therefore organic matter decomposition occurs via the bacterial channel versus the organic matter that is more difficult to decompose, where the fungi take on the role of decomposing that organic matter. And in general, it was found that legumes increased bacterial feeding nematodes, bacterivores, and forbs, so plants that have broad green leaves typically, and many of them have hardier, more sort of woody stems, increased fungivores, so fungal feeding nematodes, because the forb debris is more likely to feed fungi in the soil. The image on the right is that of Yongfu tea garden and cherry blossom in Fujian province in China. So the cherry trees seem to be planted on both sides of the road that meanders through the giant tea gardens, and when it is in bloom, it looks absolutely spectacular. But basically, in any shrub or tree orchard situation, it is very beneficial to plant cover crops, so plants of various diversity between the rows of the main crop, because shrubs and trees really benefit from the various services that other plants that can grow within the rows provide. And those cover crops can be mowed so they can increase organic matter, and there's never a competition between the tree and the cover crop, but in fact there is just a greater abundance of organisms in the soil as a result of the different plants feeding various microbial groups, and therefore increasing the overall biodiversity within the soil ecosystem, and therefore always supporting the crop, tree or shrub. And at the same time, there are a lot of pollinators that will use both the tree or the shrub as well as the cover crops. So cover crop presence actually attracts more pollinators to help with the tree pollination. So there are many benefits to having cover crops. Also, microclimates are created as the cover crops increase moisture content between the rows. So there are lots and lots of benefits to growing various plants between tree or shrub crops in order to create a more sustainable and more biodiverse ecosystem. This particular study investigated the effects of earthworms and plant residues on nematode populations. When the earthworm called Pontoscolex corethrurus was present, the total nematode density significantly increased. The presence of the worm particularly increased bacterial and fungal feeding nematodes, so their abundance, as well as density. It significantly increased fungal feeders in particular, and the reasons for this could be many, especially that earthworms distribute microorganisms as they feed and as they defecate. So there are fragments of fungi that could pass, as well as fungal spores. And then they also release a lot of nutrients as they defecate. So the castings with which they fertilize the ecosystem is a source of available nutrients. So that would increase fungal growth. It would make fungi proliferate as well as bacteria. as well as the plants that feed on those nutrients. But that nutrient stimulation and the proliferation of the fungi would therefore mean that fungal feeders as well as bacterial feeding nematodes can benefit because their food source is being enhanced by the action of the worms. In addition, this study also investigated the effects of residues, including soybean and rice, and found that these residues in general increased fungal feeder abundance. And of course, this has got to do with the fact that fungi were able to proliferate in the soybean and rice residue. Fungal feeding nematodes in the family Tillage significantly increased in buried residues, as opposed to when the soil was just mulched with the residues rather than actually incorporated into the soil. And there was no further interaction of this fungal feeding nematode family with what kind of residue was present, i.e. soybean versus rice. It was just that this fungal feeding nematode only reacted to the treatment where the residues were actually buried. There was also no effect of the earthworm for this particular family of nematodes. So as the residues were buried, the fungi were able to proliferate within the soil ecosystem where there was moisture, and the nematodes in this family obviously required those kind of conditions and that food source to be buried in the soil in order to proliferate. Aphelincus only increased in soybean residues as opposed to Ditilencus, which increased in abundance in both rice and soybean residues. In this case, there is a preference to the type of residue as to what kind of fungi grow and proliferate on that particular residue, and the nematodes respond to the species of fungi growing on the various substrates that the fungi feed on. So it is interesting to see how these different treatments, having the earthworms present and also the plant residues and the different kinds of nematodes, specifically fungal feeding nematodes, how they react to those treatments, how they react to the ecosystem when there's lots of organic matter that feeds fungi, and what kind of fungi, and whether that residue that the fungi feed on is incorporated into the soil or not, and how fungal feeding nematodes and their biodiversity is stimulated via the various ways you can actually amend the soil ecosystem. Bottom line is, as always, we need to have biodiversity in the ecosystem. And that starts with the plants that cover the soil or the crops we wish to grow. The more biodiversity there is present within the field, the more food there is for various fungal species or various species of bacteria. And that then has a flow-on effect on the biodiversity of the fungal feeding and bacterial feeding nematodes, but also they then feed predatory nematodes as well. But particularly fungal feeding nematodes are really affected by the type of organic matter that's present for the fungi that they feed on. So if you want biodiversity of fungal feeding nematodes, you must have biodiverse fungal presence, and that really depends on the biodiversity of the crops or the plants that are growing above ground, and of course the retention of that residue. It's key. So if we remove the plant residue from the soil ecosystem, that is not going to provide a food source for the fungi, and therefore our nematode populations will decline, and vice versa. So it all really comes back down to biodiverse ecosystem, starting with the plants. This image is of the earthworm that was used in the study, and there are only a couple of images on the web featuring this beautiful worm. This particular study investigated the effect of reducing tillage and how that increases fungal feeding nematodes or fungivores in the soil. In general, decreased soil disturbance and the presence of perennial crops increases nematode diversity and the percentage of fungal feeding nematodes, omnivores, and predators. Reduced tillage in this study meant no tillage or non-inversion tillage used at 0 to 10 centimeters in the different plots, while full inversion tillage digs deep into the soil to about 30 centimeters and takes the bottom of that 30 centimeters and inverts it up to the top, so severely disrupting the soil structure. So when tillage is reduced, this actually decreases the fungal network disturbance and increases fungal decomposition pathway, and therefore increases fungal feeding nematode numbers in the soil. This is because fungi grow throughout the soil in this network-like structure where their mycelium, their hyphae form a mycelium, and that actually distributes itself within the soil ecosystem. So tillage will obviously fragment this fungal network, therefore reducing fungal proliferation and therefore affecting fungal feeding nematodes. Diverse nematode community increases nutrient retention and carbon storage and creates a better habitat for fungal feeding nematodes that facilitates their movement through soil-poor water. And this basically allows nematodes to travel and also to reproduce better, as they are more likely to meet and mate as they travel through the soil ecosystem. So overall, this study found that reduced tillage increased total and labile carbon and total nitrogen and available potassium. So this led to increased microbial biomass and activity and reduced bulk density. And bulk density is a measure of compaction, how compacted the soil is. And because tillage forms usually a compaction layer at that 30 centimeter level, it actually profoundly affects plant growth and the soil ecosystem, as the compaction layer does not allow penetration of plant roots further down the soil. So it obviously creates a barrier between the topsoil and the lower levels of soil, which is really detrimental to the topsoil ecosystem, creates anaerobic conditions, preventing aeration and water infiltration. So reduced tillage in the study showed that there was a significant reduction in soil disturbance, of course, which led to increased crop residue retention and therefore water infiltration and retention, soil organic carbon accumulation, greatly improving soil structure. And it is this improvement in soil structure that creates this more favorable habitat for fungal feeding nematodes to proliferate in the soil. This image was taken from an article called Conventional versus Minimum Tillage and its Effects on Soil. And really it demonstrates just how this mechanical treatment of this living ecosystem that is soil, how detrimental it must be on it when everything's connected and flourishes in this aggregated, structured manner over time, repeated tillage, the devastating effects it actually creates. And quite rapidly, the soil is destroyed. The ecosystem just cannot withstand the constant homogenization and the amount of weight that is actually pressing the soil down as the tractor has to pass through the field. There are just so many detrimental effects to the soil ecosystem really by using tillage, and the alternative is to not plow the soil, not till, but just harvest the crops and allow the residue to drop, allow a buildup of the organic matter from the residue. And even if weeds come up, that's okay. They obviously are really good to feed microbial diversity, and over time, as the soil ecosystem recovers, the weeds will no longer proliferate, especially once the microbial populations come up to a balance with more fungal diversity in the soil. These organisms will select against weeds, and really it is about allowing the soil ecosystem to build upon itself without any further disturbance. It will take a little time, but really tillage is something that causes a lot of issues and creates really the need for the use of herbicides and other pollutants, which are unnecessary if the soil ecosystem is supported, where there is no interference or disturbance to the soil ecosystem. And instead, it is cultivated by supporting the microbial diversity within the soil. So we've come to the end of lesson 7.6. And in lesson 7.7, we will look at how, under certain conditions, fungal feeding nematodes or mycophagous nematodes might switch their food preferences to plants. So they will consume plants instead of fungi, and often parasitize plants and being associated with some severe plant diseases, while other fungal feeding nematodes may actually become predatory and feed on other nematodes. So you'll be hearing from me again in lesson 7.7. Bye for now.